Hello everyone, how are we doing today? So today we're going to be doing the exam to review for anatomy and physiology too. So just an overview of what is on this exam. Remember, it's chapters 17 through 19, and then chapter 16, which is the endocrine system. 17 is the blood, 18 is the heart, 19 is then the cardiovascular system, or all the blood vessels. So just continue with the overview. It's normal exam like we've had before. 85 questions, a mixture of multiple choice and matching and true and false. And then one bonus question as well, which on this one is a short answer unless I change it to multiple choice. But still, out of 85 points, but 90 points possible on the actual exam. And then the practical, make sure you're studying the anatomy of the heart and all the vessels for the cardiovascular system. And it's also, we did that microscope lab looking at the white blood cells. So it might also be good to familiarize yourself and be able to look at an image of a white blood vessel and be able to say which one it is. All right, let's go through this review session now, starting with chapter 17, which is on the blood. So this, blood. So this one here, make sure you know the types of cells. So types of cells include, we have the different components of the blood. So you have your hematocrit, which is your red blood cells, which are made up of the proteins hemoglobin. Remember, each hemoglobin can carry 4 oxygens. So we have our, we have our red blood cells. We have our white blood cells. I'll talk more about the white blood cells coming up here soon. Then you have your plasma, which is the the fluid portion of your blood, and then platelets as well. So plasma, where we have proteins dissolved, and then platelets. Remember, platelets are an important part in forming clots for stopping blood flow. So understand, we're talking about forming these clots now, so understand the importance of the different clotting properties. So the 2 key components in forming clots, we have fibrin plus thrombin. Well, fibrin is the actual clot that forms then. So fibrinogen is the protein. Fibrinogen plus thrombin, which thrombin has its very intricate activation process, which I won't go into here. But then these two then combine to then form fibrin, which is the actual blood clot. So if you don't have platelets, if you don't have fibrinogen, or if you don't have thrombin, you don't form a proper clot. So understand that little process right there, and that's about the overview of that process. So now we can go back to the white blood cells here. So the white blood cells can be granulocytes, or just write granular, or they can be agranular. Remember, there are 3 granular sites and 2 agranular cells, and know all the different white blood cells. So we have and know the abundance. So we have the mnemonic "Never let monkeys eat bananas," or neutrophils, lymphocytes, monocytes, eosinophils, and then basophils. Understand what each one does. So eosinophils increase when parasitic invasion occurs. Basophils are what are releasing histamine for an inflammatory response. And then neutrophils are the most abundant. Remember the N in "Never let monkeys eat bananas." And they also, if you have an extremely high neutrophil count, it is a biomarker suggesting a severe bacterial infection. And then if you do get a white blood cell count and someone is diagnosed with something called leukopenia, leukopenia is when there's a low number of leukocytes. So leukocyte is a generalized term for white blood cells. So remember, you want to understanding the different white blood cells and their importance is very important here. And then with this, be able to identify blood types as well. So know the different blood types. So we have, so A, AB, B, and O. Understand who the universal acceptor is, who the universal donor is, and how we identify each. So if I were to have a microscope slide here, and we put 2 spots of blood down, here we put the blood in red here. So there's our 2 spots of blood. And then if I'll just say this one is for testing for A. This one is testing for B. And I don't think that's the exact same white. There we go. A and B. So what you do now is you add the anti-serum to it. And if it does something called a glutinate, it denotes a positive test. So here I'll make agglutination, or this clumpiness, a yellow color. So here, if this was our sample, A type reacted, that tells you this blood type is A. So this would be A type blood. If neither of them reacted, that would be O type blood. If both reacted, that'd be AB. And if one reacted, that'd be B. So make sure you can identify what these look like on a microscope slide and so forth. So that's pretty much it on the blood. So the blood chapter was important for understanding the major components of the blood itself. And I do have that one video going over, let me open it up here, the white blood cell video that you guys have a link to. I won't play the audio to this, but here I go through and identify the different blood types. So this might be one you want to review to look at the various blood types we can find in a blood cell slide. Again, so you guys have the link to this one as well. So use it as a resource. Oops, let's open back up here. Okay, that's the blood chapter. Now let's go to chapter 18, which is the heart. Now this one you might have to spend a little bit more time studying, because you do need to know, of course, the anatomy of the heart. And there's a lot to the anatomy of the heart. You have all your 4 different chambers. You have your right atrium, left atrium, right ventricle, left ventricle. So make sure, again, I'll shout out this little lab video. Hope that's the wrong one. I want to use the one that I made and edited. So that's this one right here. So make sure you review this video here, where I label all the different parts if you forget what they are. Now note that in this video, I did label the pulmonary trunk here, which is technically the pulmonary artery. I wrote vein right here. In the video, I did say artery the first time I was mentioning this, but then I wrote vein. It happens. But make sure you realize this is the pulmonary artery or pulmonary trunk, not the pulmonary vein like I wrote there. I should put a big X on that. But review this video for the anatomy of the heart. I then go inside the heart here, looking at the major structures of the cusps, not the cusp, but the valves. So the mitral valve, bicuspid, tricuspid valve, things like that. I label them all here. The chordae tendineae, the papillary muscles, and so forth. So a very important little model here going over. And then here I go over the conducting networks. So the conducting networks are also very important here, understanding the pathway of conduction through the heart. So from the SA node to the AV node, down through the AV bundle, through the bundle branches in the subendocardial conducting network, or those Purkinje fibers. Sorry if the captions are hard to read on this video right now. I kind of designed them to work best in OneNote here. But now they are back. So yes, the anatomy of the heart is a big section of the heart chapter. And then also direction of flow. So know the order in which they come in. So, talking about coming from the lungs, coming through the pulmonary arteries, and how then it gets pumped out through the rest of the body, or are you returning from the body via the superior and inferior vena cavas? So keep that in mind. Understand the flow of heart. I know we focused on that in class 2. And then talking about the nodes. So here, nodes, we have the SA node and we have the AV node. AV node leads to something, if this breaks down, it causes what's called a heart block. And this AV node is also the key pacemaker of the heart. If the AV node fails to fire, you don't get any ventricular contraction and you get no blood flow. But SA node is a sinoatrial node, but this one fires first to contract the atria enough to pump the remaining little reservoir of blood into the ventricles. And then with these, understand their rules and what happens if you are having something like arrhythmia, which is irregular heartbeat. So understand the rules of the pacemaker cells, how they work via a process called automatic depolarization. Now you don't have to know the details of the ion movement and things like that there, but understand what automatic depolarization is and what that means. So that's understanding how they work, and then also how they get reset. So we use something called a defibrillator, which stops them from firing, and then we refire them, and it should reset the heartbeat. So understand that little process there and be able to explain it. And then, like I said, understand that conduction network through the heart and how that signal passes through the different regions. And then make sure, because you're going to be reading questions, so make sure you know the difference between diastole versus systole. So, relaxation, relax, contract. Because questions might be worded via, what happens during diastole of the ventricles. So if you don't understand what diastole means, you don't know that I'm asking what happens during the relaxation phase of the ventricles. Not that that's a question, but just giving an example of using that in a sentence. Then remember, there's a difference between the left and the right side of the heart as well when it comes down to the size of the myocardium. So remember, the left ventricle is larger and thicker because it has to pump blood to the systemic circuit, whereas the right ventricle is actually smaller because it only has to pump through the pulmonary circuit and doesn't require as much pressure. Next, the ECG or EKG. I write ECC, or the electrocardiogram. Understand this wave. So if I were to try to draw it here, so we're, coming down through, there's the graph. Remember, this represents the combination of contraction and relaxation of the action potentials of the heart at the same time. So as we're coming down, we go through this little bump right here, jumps down, jumps a big bump up, comes back down. I know this isn't perfectly drawn, comes back up, and then through here. So right here, we have, P, Q, R, S, and T. So understand the important parts of this. So this would be time, and then this is voltage, I guess. This is voltage over here. So voltage versus time. And you're looking at the total voltage change inside and outside of the heart as this action potential moves through the heart. So something like this part here, the P, this is where, atrial depolarization occurs. So the atria are contracting. So this is atrial systole, contracts. And then we get ventricular contraction as we move up. And so if we're looking at the point in which pressure begins to rise in the aorta, it would be past this point here, because this is when you're getting ventricles contracting and then pushing that blood into the aorta. And then here, this last little bump here, or the T segment, this would be when ventricular repolarization is happening. This is also when blood exits the aorta here. If we're drawing the aortic valve right here, this is actually when blood pushes back on that valve, and that valve closes, and that's that dup sound we hear when listening to our heartbeat. That's actually the blood pushing back down onto that closed valve. So, the second sound made by the heart also happens at this little end wave right here. So understanding this wave here is really important. I know we drew that out in the laboratory as well, so look at that again and make sure you understand the different components. All right, that is the blood chapter. Next one I want to go through is the vessels, or are the vessels. So here, chapter 19, I'll just call it vessels. Again, the big one here is understanding all the different anatomy of the vessels. So know the major vessels, know where they go, and know how to name them. Again, I did make that video for you guys that you can look at now. So here, looking at the different vessels and the layout on the model we have in the laboratory. And just as a note here, when I was talking about the draining pathway of the arm, here, let's get to the arm draining. What did I talk about? Right here, I called this region right here the brachial vein. It's actually the axillary vein. So this is the brachial vein coming in here. This is the basilic vein coming in here, and this is the cephalic vein coming in here. Where these two come back together, this is the axillary vein. And then through here then is the cephalic vein, and right here it's called the subclavian. So just one thing I wanted to point out in that one. And down here we also have, remember this one's the median cubital vein. This is where blood is typically drawn. And then also the radial artery is typically the site where we take pulse. So understand these important veins. So right here, radial artery, right there is an important site for pulse because you can push against your radius, and then you can feel that pressure gradient as you push through that region. So a few things you want to keep in mind there. So the anatomy and the key blood vessels and so forth. There are a few matching sections in your virtual lab that highlight some of the important ones. So you want to look over those as well. And then with the vessels, we did talk about capillaries. So here, the capillaries, there are three main types of capillaries. There are continuous. Continuous capillaries are what we find in the blood-brain barrier. So they're kind of restricting the amount of flow that comes through. Then we have fenestrated capillaries, which allow a bit more flow. So there's more fenestrations, or we talked about that Swiss cheese. So here, fenestrated. And then the largest space capillaries where a lot of flow can come out are the sinusoidal capillaries. So make sure you understand the different capillaries and how the pressure affects the capillary bed. So capillaries spread out. So we have, one part coming in, and then it spreads out into many, many different capillary beds, and then they all come back together in one. So this spreading out of this blood as we move through not only slows it down, so it also reduces the pressure a little bit as well. So it allows for more time for diffusion to take place, and that slight pressure in there allows diffusion to happen as well, and it protects the capillaries from exploding. You don't want the capillaries to be too high of a pressure because they are weak. So we're going to keep that in mind too. And then when blood is flowing, pressure does change throughout the circuit. So it first pumps out the heart. So pressure is actually highest in the arteries, and we go to the arterioles, and we go through the capillaries, and then we go to the venules, and then back up through the veins. So in the veins, we have the lowest pressure, and that's why in the veins we have valves that prevent the backflow of that blood. Understand the short-term versus long-term regulators of blood pressure. So short versus long-term blood pressure, or BP, regulation. So short-term involves the baroreceptors and chemoreceptors that are causing vasoconstriction and vasodilation, increasing respiratory rates, decreasing respiratory rates, and so forth. So those are short-term. Also, so those reflex arcs from like vasomotor fibers and whatnot, monitoring the actual blood vessel stretch and so forth. Now, long-term then includes major alterations to blood volume, and those are done via that renin-angiotensin-aldosterone pathway from the kidneys. We'll talk about that more when we get to the urinary system. That's typically the one way we can represent the long-term regulators. And then with blood flow, understanding the properties of blood viscosity. So if blood viscosity increases, you increase what's called peripheral resistance. So it actually makes it harder to flow blood, and that could be dangerous if you get a high hematocrit concentration. Increasing your blood viscosity increases your peripheral resistance and reduces the flow rate. So we use the straw analogy when talking about that. So it's important to understand that too. Let's see, what else do we have here? That's pretty much it with the blood. The big thing is understanding the different vessels, and that will be the hardest part in studying the blood chapter. Then the last chapter here, we have chapter 16, which is endocrine. Now, I did make a little video. I'll show it again here. It seemed to work well for the other parts when discussing them. So here, the endocrine system, going over the major components of the endocrine system, the major glands, and so forth. So you want to understand the general overview of where we can find these in the endocrine system. I mean, there might not be direct questions on the practical about them, but I might have a matching section on the exam regarding the important components of the endocrine system. So you definitely want to check out this video as well as a little review here. So that would be important too. Then I know this is going to be the hard part, but all hormones that we talked about. So understand all the hormones. So what their function is, what they do, their importance, and so forth. So just to go through a few of the examples. So something like the thyroid hormone, that one's important for growth and development, maturation of the nervous system, and iodine is an essential component to form those thyroxine hormones. Remember, we talked about that formation process. You don't have to know the details of that formation process, but that's something that iodine is very important for in our body. Then the hormone leptin is something that suppresses appetite, increases energy expenditure. Melatonin is important in your sleep and wake cycle. Growth hormone, important for development and height, so you don't produce enough growth hormone, you don't grow as tall. Aldosterone, the renin-angiotensin-aldosterone pathway that I just mentioned. So aldosterone is important regulator of salt and water balance. And then to go with that, we have antidiuretic hormone, which decreases urinary output and dehydration, and that can also increase blood volume. Another example of a long-term regulator right there. Then you can have glucagon versus insulin. Glucagon raises blood glucose, insulin lowers blood glucoses. Now, this is just naming a few that I decided to throw down on a list here, but there were others that we talked about. So, have a good understanding of all the hormones. There's a good matching section in a virtual lab, so make sure you understand the basics of all these hormones. And also remember, direct versus indirect. So the effects a hormone can have on a cell and how it has to work via gene expression. So here for gene expression, hormones are usually causing gene expression. So direct means it can go through the cell membrane directly to the nucleus and cause gene expression. Indirect means it needs to have a second messenger. So a second messenger system such as cyclic AMP or that G protein coupled mechanism we talked about. You don't have to know the key steps to the G protein coupled mechanism. Just know that that is referring to an indirect mechanism where that hormone can enter the cell, so it has to send its message and have a second messenger to send it inside. And then I know you're going to hate this part, but the layers of the adrenal gland. So you have the adrenal cortex, the adrenal medulla, and we drew it in our workbooks, and also we looked at the image of where it's in the virtual lab too, is what I was trying to say. So make sure you understand what's produced where, and so what's produced in each layer. Very important that we understand the different layers of that adrenal gland. It has the outer layer and the inner layer as well. So like the gonadotropins, where they're released, the adrenocorticotropins, things like that. And you'll review those and you'll be okay. And there's that one question from the virtual lab you'll want to review there. And then the different diseases we talked about related to the hormones. I should spell diseases correctly here. Diseases. There we go. Understand various diseases related to hormone imbalances. So like diabetes is a hyposecretion of insulin from the pancreas, of course. Addison's disease is a hyposecretion of the hormone in the adrenal cortex. Graves' disease is an autoimmune disease of the thyroid. Acromegaly is hyper secretion of growth hormone during adulthood. Pituitary dwarfism is hyposecretion of the growth hormone during childhood. So you also had a matching section in the virtual lab with the different diseases and so forth. I'm not going to write them all out here, so make sure you review that as well. So I know this was a shorter review video, and that's because I have other videos to now go with this. I didn't have those before, but we'll be slowly making more videos as we do this, and these reviews won't be as long or lengthy because we have the other sources to look at. But that's all I have for this one here. If you have questions when you're studying, feel free to reach out to me, check it out, and so forth. But with that, I hope you all have a great day, and you're all going to do great on this exam. And yeah, there, motivational speech. All right, I'm going to go upload this now, and bye-bye.